Hello everybody, my name is Gwen Knight. I'm an Associate Professor and Co-Director of the AMR Centre at the London School of Hygiene and Tropical Medicine. Welcome to today's workshop, which is entitled New Perspectives on Bacterial Drug Resistance, and it's organised by the JPIAMR. I'd like to thank everybody for joining, and thank you in advance, in particular to the speakers for preparing for this event. Just a reminder to everybody that this is being recorded. If you have any questions, please post them in the chat. And a reminder to everybody to remain muted with your videos off unless you're called upon. So hopefully during the discussion sessions, we can have unmuted with video conversations, but please do wait until you're called upon before unmuting yourself. So the purpose of today's workshop is really to try to identify key priorities, challenges, and opportunities for research in the topic of antibiotic resistance. And that's really to inform and to define future priorities for funding bodies. So what we wanted to do today was bring together some new perspectives and new insights from early and mid-career researchers working on antibiotic resistance to try and understand where the field should go in the forward, where are the gaps, and where we think we can come up with new ideas for what funding bodies should be putting their money into. So the aims of this workshop are therefore fourfold. So the first one, we're aiming to identify existing knowledge regarding antibiotic resistance. And for that, we've got 4 keynote speakers who are going to speak to specific One Health topics. So we're hoping that these are going to be somewhat new insights. So these are not the names you've heard speak before. They're going to give us fresh perspectives on where they think the research is going in their particular field. So we're going to hopefully hear from somebody talking about policy and national action plans, and then think about human health, animal health, and planet health. Now, this is not I aim to be exhaustive to cover all aspects of One Health. We just want to give some nice highlights of important areas for consideration. So that tackles our first aim to identify existing knowledge. So the second aim is really to try to identify knowledge gaps for further research. So after hearing these keynote speakers, we'll have a 15-minute break. And then when we come back, we'll hear perspectives from 6 early to mid-career researchers. This will give us a broad range of ideas on what potentially is needed, and these are sort of aimed to be specifically targeting and emphasizing gaps. The third aim is to consider the policy implications of bacterial drug resistance, because ultimately antibiotic resistance is a public health issue, and what we want to know is what can we do with this knowledge to go forward to prevent future disease burden. And so we'll do that by thinking about where the gaps are and how those gaps could by filling those gaps, we could do better at evidence-based policy decision making. The final and fourth aim of today is to identify and prioritize future research priorities for bacterial drug resistance. Hopefully we'll identify those as we go through both our keynote speakers and our early and mid-career presentations, but we'll also have a half-an-hour session at the end where we can pull together some of those cross-cutting themes and the main opportunities for future research funding focus. And this will be the opportunity for the non-speakers, so the other participants, for you to ask directly questions and to you to raise potentially ideas and thoughts that have come to you during the speakers that we hear today. So I'm now going to pass over to Caroline Johnson from the UKRI MRC, who's going to give us a brief overview of the JPIAMR SRIA. So Caroline, over to you.
Thank you, Gwen. So I'm just going to share my screen super quickly. So just tell me if this is actually the right one, because you know what it's like. So hopefully that's the correct screen. So hi everyone, I'm Caroline Johnson and I'm the Programme Manager for the UKRI MRC. And I'm here today actually to introduce the Joint Programme Initiative for Antimicrobial Resistance, or JPIAMR for short. And they're organising this workshop as part of a larger series of events. So first of all, I'd really like to thank you all for taking part in this workshop to take a fresh look at our understanding of bacterial drug resistance and to see what gaps remain. So I'm just going to give you a really super quick introduction to the JPIAMR and this workshop, which, as I mentioned, is part of this larger consultation to work out future priorities for research and innovation in the field of antimicrobial resistance. So as you can see from this slide, JPIAMR is a global organisation bringing together 29 countries with the common goal of tackling AMR in a coordinated manner. Part of the goal of the consultations that we're running at the moment is to update the strategic research and innovation agenda of the JPIAMR. So currently this covers research and innovation for both fungal and bacterial resistance, with 6 priority topics which range from intervention through diagnostics and surveillance. To date, JPIAMR has invested more than 127 million euros to fund 99 research projects and 38 networks. Funding's been given out to more than 1,400 researchers from 77 different countries, including 117 researchers from lower and middle income countries. So you can really see that there's a global reach of the JPIAMR here. And here you can just see a little bit more about the distribution of the JPIAMR funding across those 6 priority topics, which I thought was something really useful for us to understand for today. So as we can see, to date, funding has been fairly evenly distributed between transmission, therapeutics, environment, and interventions, where diagnostics and surveillance are lagging a little bit behind, but there is a network call open in this area at the moment. But this is just something to bear in mind as we kind of go through this workshop, where we have funded and where we haven't funded to date. So just to kind of sum us up, if you want any more information about JPIAMR, please check out their website and social media. And I really hope you enjoy this workshop and get a lot out of it. And I'd like to give a really quick thank you to our chair and our speakers for agreeing to take part in this event. So yeah, thank you. Oh, and I will stop sharing now.
Thanks, Carolyn. It's good to see that that overview was quite split though, the 20% in each of those, but yeah, and the diagnostics one, that will be bolstered by the newest call. Great. Okay, so without further ado, we're going to launch into our short keynote section. So I'm very happy to introduce Chantal Morel from the University of Bonn. So Chantal is a health economist specializing in infectious diseases and in issues related to antimicrobial resistance in particular. So Dr. Morel is involved in estimating the cost of antimicrobial resistance and in the comparison of AMR-related interventions on an economic level, for example, via cost-effective analysis and cost-utility analysis. So Dr. Morel, I believe you're going to discuss the implications of policy and national action plans in the context of AMR for us now. Would you like to share your screen? Great, thank you.
Oops, I think I need to go.
And just, yeah.
Maximize. There we go.
Great. Just a reminder, you have 10 minutes. I'll do a wave at 8, so you can start wrapping up. That's okay.
Okay, great. So hello, everybody. Just to say a couple things quickly. One is that I hope I can make it within the 10 minutes. Also, I haven't timed this presentation, so I'm not sure exactly how long it is, but I'll do my best. Another thing is that I'm a health economist, as you just heard. So my take on policy is from an economic perspective, just to sort of predicate this on that. So you just had my profile, so I'll just skip my first slide. I'm going to be focusing on 3 main critical challenges. The first having to do with the limited implementation of national action plans within the lower income countries. The second having to do with the limited support for efforts to slow the growth of AMR at global level, which also relates to limited support action plans at country level, as well as limited progress on the side of investment for AMR-related technologies, which also is related. This is kind of a lot to go through in 10 minutes. I realize it's probably a little bit too much, but let's give it a go. Sorry, I'm having trouble. There we go. Okay. So the first one, national action plans within mainly low income countries. So most national action plans have components. I mean, there are different types of components that have been chosen by different countries. They generally include an antibiotic stewardship component, an infection prevention and control component, surveillance, education and awareness, and cooperation. And there's a few other variations of that, but generally they have these 5. Now, many countries have had their NAPs agreed upon, over 5 years ago. A lot were basically made official and also approved at WHO level in around 2017, 2018. So it's been a little while, but not very much has happened on the ground in many of these countries. Now, why is AMR not a priority in these countries? Well, as we know, AMR is not a disease. It's not always easy to understand what we're talking about when we're talking about AMR. It has no distinctive set of signs or symptoms. Also, within most of these settings, we don't really have an AMR champion to really, hammer home the messages with regard to AMR. And then finally, there's not that much evidence from the ground on the actual impact that AMR is having. And I would stress that in particular, there's no evidence of what it's costing these economies to have AMR increasing. And what happens then is that the ministries of finance can't really gauge where AMR belongs amongst all the other pressing priorities that they have. And the result is that we don't see very much movement on AMR. Now, this is just to illustrate what happens when we don't have much evidence of AMR on the ground. So basically, we don't end up prioritizing AMR at national level. And then when funders see that, so mainly like development agencies and such, when they see that AMR is not a priority, they're much less likely to fund AMR efforts on the ground. So then you end up with this cycle of there being no funding to explore how AMR is really affecting society. Then you don't prioritize it, then there's no funding, and it just goes around in this sort of vicious, evidence action cycle. And that's what we're seeing in many of these countries. Now, just to give a quick blurb about what's going on in terms of the funding of the NAP implementation right now, initially the tripartite agencies of WHO, OIE, and FAO put together the multi-partner trust fund for AMR. They were then joined by UNEP, so now it's the quadripartite MPTF for AMR. This is meant to be the UN's main mechanism to fund NAP implementation, but it's really not, and there have been a handful of countries that have received some funding to push forward on implementation, but this is not a lot of money given what we're trying to do in the NAPs, given what the countries have signed up to do. It's really not a lot of money. And well, effectively, it's somewhat symbolic. So the MPTFs are really a catalytic fund. It's mainly to stress that we need to be trying to tackle things, in a One Health manner. So looking across all sectors, but it's not really going to be, we can't count on the MPTF to really be funding full implementation of NAPs. We need to get funding from elsewhere. One hope is that Global Fund, now with its new budget, that's supposed to be, I think they're going to be spending, I think it's 1/3 of their budget on pandemic preparedness. So there's some hope that we could be getting some more funding for NAPs through that, but that's really unclear at this point. And then there's World Bank. And World Bank, the way World Bank works is that countries basically establish their priorities express their priorities to World Bank on these two main documents. They're sort of 5-year documents. That's the Systemic Country Diagnostics, otherwise known as the SCD, and the Country Partnership Frameworks, the CPFs. And you set about your priorities for the next 5 years, and then you can make a funding application based on those priorities. But if you haven't already established AMR as your priority, they are not going to fund AMR related activities. So you've really got to be clear on what your priorities are if you want to get funding through these various routes. Now, I'm just quickly throwing this out there. So my team right now is setting up a project in Zambia and one in Malawi, trying to cost AMR at country level, such that we can try to get some evidence for these funding applications and to try to motivate action within country, including within the Ministry of Finance. We're going to be using the framework set up by the GAP1 network, which is a huge network that basically tried to come up with it's somewhat of an ideal framework. If we were to have all the data that that we could imagine, what would we need to do a proper costing of AMR in country. So we're using that framework and now trying to operationalize it within Malawi and Zambia, looking across all sectors. Now, I think probably most countries are going to need to do some sort of an exercise that, to make an investment case at national level as to why we need to prioritize AMR. So just in terms of recommendations, I think we need to do some sort of an exercise within each country. There should ideally be some variation in the estimates, especially for transmission, such that we can try to model and see what these costs look like if we were to have some sort of an outbreak. And then we really need to be supporting cooperation across these countries to undertake, when they're undertaking these costing exercises, because there's so much work involved, but we really could, we don't need to be reinventing the wheel every time. A lot of the thinking can be collective, and the countries can be, learning from one another, doing, throughout these exercises. Second issue, so at global level, thus far there has been limited support for efforts to slow the growth of AMR, including the implementation of national action programs. So beyond these little, bits of development aid for AMR and some verbal assurances, at the level of the G7 or whatever, the richer countries have done relatively little. Now, to my mind, this is only going to change if we see the economic implications of inaction within these higher income countries. And I think that, basically, there needs to be a case for self-interest for these higher income countries. And it's absolutely there. And we can see from COVID-19 that it is there. And I think we should be capitalizing on what COVID-19 has given us. So it's given us a lot of real world data from across all the sectors of the economy of what it looks like to have an outbreak of infectious disease, and how much it has cost these economies. And importantly, okay, so just to say that, we are now in a time when, the estimates of how much an outbreak of AMR costs society, we have real data now. So these are no longer just estimates that, can be brushed aside. COVID-19 gives us real data. So it's not at all what we were facing before. Sorry.
Chantal, you're just running out of time now. 30 seconds.
Okay.
Can you wrap it up? I know.
I'll wrap up on this one. Yeah, these are really the two most important anyways right now for this talk. And importantly also, this is there's a very useful parallel here in that COVID-19, just like AMR, was not a disease in and of itself. It was the worsening of an infection that we already knew. So in some ways, it makes it easier to then internalize AMR because it is really quite similar in that respect. And I think we should capitalize on that familiarity that COVID-19 has brought us. So a recommendation, we should conduct economic analyses of AMR in a few key countries, high income countries, using the evidence from COVID-19. And I think it's important that they're at the national level and not international level, because it's much easier to build political support in country that then can be sort of translated to support for efforts abroad as well. But building that political support is really important, and that doesn't tend to happen when you make an estimate just at global level. And I'll stop there.
Great, thank you very much, Chantal. Does anyone have any clarification questions for Chantal? I don't think any have come in the chat, Chantal. So in interest of time, we'll just move on, and then hopefully you can join us for the discussion later. But if you do have questions for Chantal, you can put them in the chat, and hopefully she can answer them there. That was really interesting. It's great to see you talking about the funding model and how that really drives whether national action plans get implemented, because I think we've seen that with other infectious diseases where it's really the priority setting, even within, not just nationally, but within the global fund, how countries can access that money in order to implement what they need to do on the ground. So hopefully there should be some cross-learning from other diseases for AMR. Okay, let's move on to our next. We're switching direction slightly, but we're still thinking about human health. So it's my pleasure now to introduce Fernanda Paganelli, who's an assistant professor at UMC Utrecht and a coordinator of the JPIAMR funded European Consortium MR. And is also part of the Dutch One Health programme to study the resistome in humans and animals. So Dr. Paganelli will be discussing the problem of AMR in the context of human health. So over to you, Fernanda. I can see your screen. Thank you.
Thank you very much. It's a pleasure to present here today. And as Chantal, I just want to give you a general overview what we see here, how AMR is affecting human health, and what should be the future approach and where are the gaps. So of course, we know all here that AMR is a global health challenging. And if we make a comparison with other pandemics, like SARS-CoV-2 that Chantal just mentioned, actually AMR is emerging as endemic problem everywhere, but it's always under radar. Like she said, it's very difficult to quantify and really study, but that will explode in a point and can deprive our future generations from treatment if we cannot control it. So in our opinion, the development and implementation of efficient interventions will be the way to go. And these interventions should control the emergence, so the selection and transmission of AMR. However, the old, let's say, old-fashioned way, or most of the studies have been focused on using antimicrobials, what we know can actually increase resistance over time, and also improve hygiene process to prevent the AMR spread. However, we see AMR as much more complex and multifactorial problem. So to have really efficient interventions and really future perspectives to control AMR, we need to look at it in a more complex way and more multifactorial way. And nowadays we lack the knowledge about the dynamics of antibiotic resistant genes and antibiotic resistant reservoirs, not only in a hospital setting, but more in the general whole ecosystem. So abiotic and biotic surfaces, and also relation to animals and the environment. And we really have to have more clues about risk assessment analysis in these different settings. And one thing that we already know, that once you have antibiotic resistant bacteria, they are select and widely disseminated in these, that's called eco-evo. You need eco-evo interventions to restore and to have more of susceptible bacterial populations in humans and animals. And another thing that is also being more and more evident is that once you have health microbiotas, and this is not only in humans, but in general, in animals and ecosystems, you usually can control better or have less dissemination of AMR. So once we think about a complex system, one word and one topic that comes back on and on is this One Health. And in principle, this means the risk assessment of emergency, transmission, and maintenance of AMR in this interface between humans, animals, and other linked environments. So the selection and transmission events in the human health or animal health usually happens in what we call AMR hothouses, so hospitals, households, and farms. And actually controlling these different environments would be a way to control AMR spread. So we see as interconnection between all these different environments, let's call like this. So where you have indoor microbiome communities, what would be like in hospitals, and in farms, for instance, to outdoor. So that you also contaminate, for instance, the community and the environment, for instance, wastewater. So how do we see as possible ways to tackle this problem? And here I divided in two ways that the interventions could work. And one way, like I mentioned previous, is interventions to reduce or the selection based on the microbiome preservation. So we know that the microbiota in the health humans, as in patients, can really have a big role in colonization resistant against antibiotic resistant bacteria, and also preserving these in the environment can be a way to prevent dissemination and transmission of AMR. So nowadays, different approaches, let's put like this, have been considered in terms of intervention. So, for instance, biodegradable antibiotics, or compounds that can inactivate or absorb antibiotics, bacterial phages cocktails that you can directly target a specific antibiotic resistant bacteria, and fecal transplantation, so that you really try to restore the diversity when you have sick patients. Another part of the intervention is actually try to reduce transmission in a different approach. So, for instance, developing specific drugs to reduce dissemination of AMR, or more social measures to prevent to break the ecological connection between these different niches. And this is what would the really One Health approach being applied. So try to avoid the transmission between animals, community, and patients. But of course, this is not so simple. And I think we have a long way to go, because there's still very little that has been done so far. So when we talk about microbiota preservation, microbial preservation, there is almost no applications, for instance, for fecal transplantation for MR. The most known example is in Clostridial diffuse infections, so that it was shown that can control the infection is a way to do it, but not specifically for MR, for specific multi-drug resistant pathogens. So we needed to create ways to study better and understand better how this approach could be used in patients that we have concerns about colonization with multi-drug resistant pathogens. When we look more the outside, so in farms and wastewaters, there is an increased report that these are big reservoirs of antibiotic resistant bacteria, genes, and mobile genetic elements. And usually these can cause outbreaks that in the end can also put in risk patients that are colonized, and as everything ends up again in the population, in the community. So understanding better how these different complex systems are linked is really important to have direct interventions. Next, also think about outdoor microbiota. So we know nowadays that airborne transmission seems to be really important in some hotspots, and the ways that interventions could influence that in urban spaces, or for instance in farms, is really important and have been evaluated in depth yet. So in summary, we have, in my opinion, a long way to go, but there are a lot of options and different ways to explore. And more we are seeing this as a multi-complex problem, and not only a problem that you would see in the hospitals when you talk about human health. So we need much better diagnosis and surveillance to implement and monitor these interventions, and to really measure the impact and efficiency. And in the same way, influence local conditions. So the local epidemiologic part, so as Shantan also mentioned, like the local regional applications is important, but also when we talk about in specific environments like farm or specific urban areas, and that we lack actually tools and platforms to analyze all these metadata. And I think that was it. Thank you. If someone has questions.
Thanks very much, Fernanda. If anybody has any questions, just pop them in the chat and I can ask them to Fernanda now. It's really interesting. As you say, it's really important to highlight, I think, the fact that AMR doesn't mean removal of all bacteria, right? We should be using the microbiome as a way of supporting and out-competing resistant bugs. Is there such a thing as a healthy microbiome as a built environment then? Is that where we're going?
Yeah, exactly. I think that we can really translate to the environment, right? And we understand much more about ecology in a bigger setting, but not too much about microorganisms yet. What is surprising, because so many people work nowadays with microbiomes, but I think we're still in the infants, how to really use it and understand the role, especially when we talk in relationship to AMR.
Yeah, really important. Great, thank you very much, and thank you for sticking to time, Fernanda. I'll pass on now to Dr. Mattea Pirolo from the University of Copenhagen. So Dr. Pirolo is a PDRA from the University of Copenhagen, working on multi-drug resistant bacteria of both clinical and veterinary relevance. He's involved in international research projects aimed at reducing antimicrobial consumption in pig production and at developing new diagnostic tools to rapidly identify AMR pathogens in both companion and farming animals. So Dr. Pirolo will be discussing the problem of AMR more in the context of animal health, so shifting our focus slightly. Over to you, thank you.
Hello everyone. I hope you can hear me, and thank you, Gwen, for your nice introduction. So yes, I'm really glad to be here to present some perspective on bacterial drug resistance, especially in animal health. So I'm Mattia, and I am currently employed as postdoc in the One Health antimicrobial resistance group of the University of Copenhagen. And for my presentation for today, I would like to talk about some perspectives which are more or less in line with the focus areas of our group, and our group aims at improving diagnosis and antimicrobial therapy of bacterial infection in animals, especially using a One Health approach. So for the sake of this presentation, I divide the perspective of AMR research on animal health in two big groups, as you can see from this graph. On one side, we have the improved therapy, which aims to minimize the selection of antimicrobial resistance by optimizing the use, the dose, and the duration of the antimicrobial therapy. And from the other side, we have the improved diagnostic, which will ultimately allow for a rapid and identification, but also antimicrobial susceptibility testing of a bacterial pathogen. And here I've highlighted some examples in these two strategic focus areas. Within the improved therapy group, I just brought some example about alternatives to antimicrobials, but also intervention to reduce the antimicrobial usage and its consequences. Whereas in the other big focus areas, the diagnostic area, I highlighted some examples such as the point of care testing and the clinical breakpoint. And finally, in the as you can see here in the middle, I've also highlighted the prognostic biomarker, which I will talk to you about a bit later. So regarding the improved therapy and alternative antimicrobials, in 2018, the European Medicine Agency has classified antimicrobial for use in animals in 4 different categories, which are highlighted here on the right. And so we have category A, which are antibiotic to avoid, category B, which is basically antibiotic restricted for animal use, and then other categories. But in general, the principle of AMA is to promoting a prudent approach, a prudent use of antimicrobial in animals. And of course, this means that antimicrobials in animals are actually limited, and that alternative to this antimicrobial are not available for all disease conditions. So in this context, in this framework, we initiated a European Innovation Action Project called AVANT, that stands for Alternative to Veterinary Antimicrobials, that focuses on pigs which are the main species in which animal species in which most antimicrobials are used. And more specifically, we are focusing our attention on post-weaning diarrhea, which is the main indication for antimicrobial use in pigs. And it's extremely important given that an alternative to treat this infection are lacking, especially after the restriction imposed by the EU on colistin and zinc oxide. And here on the right, you have the Avant portfolio with all the intervention that we are currently testing. And so we are testing gut microbiome modulators, including pre and probiotic, but also fecal transplantation, and some specific compounds that target the ETEC, the so-called ETEC, or enterotoxigenic E. coli, which are the main cause of main pathogen associated with post-weaning diarrhea. But within this portfolio, we also have immunostimulant and feeding strategies. And regarding the second point of the improver therapy focus areas, we also have a project founded by JPIAMR called FarmCare, that stands for Farm Intervention to Control Antimicrobial Resistance, which aims at promoting and trying for intervention to reduce the antimicrobial antibiotic use and the consequences. And we are doing so with different intervention by controlling the AMR spread by avoiding mixing and remixing of piglets, but also by preventing the spread of AMR gene to human through improved farm biosecurity. And finally, we also have two different approach, a machine learning approach to identify high-risk piglets that will develop post-weaning diarrhea. So for early detection of high-risk piglets. And these two approaches are, one of them is based on a metagenome approach that will hopefully ultimately identify some prognostic biomarkers. And this is why I mentioned prognostic biomarkers at the beginning of my presentation. So in general, prognostic biomarkers indicate an increase or a decreased likelihood of disease recurrence or progression. And generally, this should be easily measured and should be used as a benchmark for prevention and control of disease. And the metagenomic analysis, in particular the microbiome analysis, are really hotspot for identify prognostic biomarker, because microbiome structure that are potentially associated with chronic disease can be used as biomarker to predict the disease progression. And in this context, we have a project where we analyze the microbiome structure of the respiratory tract in pigs, and in both healthy and healthy animals and animals which present symptom of the so-called PRDC, or porcine respiratory disease complex, which is a critical respiratory syndrome in pigs, but also a common reason for antimicrobial use in this animal. And our preliminary results that come from our study, where we analyzed the structure of both the upper respiratory tract, but also the lower respiratory tract of healthy and diseased animals, help us to identify some taxa, which are significantly associated with both sick or healthy animals. And this we could represent microbial signature that associated with the animal health status, and hopefully they will become in the future prognostic biomarkers. And I will also, for the sake of time, I'm rushing a little bit here, talking about also, I would like to talk about the other part of the focus area, so the improved diagnostic. More in particular, I would like to talk about the point of care testing, which is a medical testing that takes at the time and place of patient care. And ideally, point of care test should provide much faster access to results compared to the traditional practice of sending a clinical specimen, so a clinical specimen to analyze to a diagnostic lab. But this is only an ideal world, because as probably you know, point of care testing are for infectious disease in animals are less developed than in human medicine. And in this context, we also have this is just an example, a project of to guide antimicrobial choice for treatment of pyoderma in dogs. More specifically, we are targeting Staphylococcus pseudointermedius, which is the major cause of pyoderma in dogs, and also a common indication of antimicrobial use in the smaller animal veterinary practice. And our test that we're developing now is based on a simplified version of a PCR that is called LAMP, or a loop-mediated isothermal amplification. And this test allows for a rapid and direct identification of staph pseudointermedius, but also to the resistant gene that for the first line antimicrobial. And this test basically allow to identify again staph pseudo and the resistant gene in more or less 1 hour from more or less all clinical specimens, including swabs from cutaneous lesion. So I hope that I give a more or less comprehensive overview on our focus areas and some perspective on AMR in animal health. And I would like to invite you to follow us on social media, because we're really active where we post our latest results. We do have both a Twitter account, but also an Instagram account. And I'm, of course, I'm looking forward for the discussion on this perspective. And I would like to thank you all for your kind attention.
Thank you so much, Mattia. It's very interesting. We haven't got any clarification questions in the chat. If anyone has any, please do pop them in. Interesting that the microbiome has come up again, this idea. I was wondering how rapidly you see changes, the changes that you've seen. Is it do you see them in the days or the hours before the disease onset, or is it weeks? How long do you have to actually look for these, or you don't know yet because it's not prognostic?
Yeah, that's a really nice question. And what we have done so far is a pilot study, which is a cross-sectional study, and we do observe differences in the microbiome structure. And of course, longitudinal study will more or less be ideal in this type of to identify prognostic biomarkers, and we are actually working on that as well.
Great. Okay. Thank you very much. Okay, in the interest of time, I'm just going to pass now to our final speaker. So our final speaker in this first session is Dr. Edward Haynes, who's a molecular biologist at Parascience. He's working on the genomics and epidemiology of bacterial disease, and Dr. Haynes is currently part of the sequencing team at Parascience studying plant pathology and targeted detection of novel viruses and the genomics of bacterial and fungal pathogens of plants and honeybees. So Dr. Prello is going to move us again from animals now into the context of plants. So over to you, Dr. Haynes.
Thanks very much. Has that shared okay to the screen?
Yes, we can see you and see your slides.
Excellent, great. Well, thank you very much for the introduction, and thanks for the invite to talk to you today. Conscious that we're a little over time, so I'll try and dash through this as quickly as possible. So I'm going to give a kind of broad overview of antibiotics and AMR in plants and in plant health. It is a broadish overview, but it's based on a review that we were commissioned to do by both FSA and DEFRA a couple of years ago, which is still relevant. And I think, so I'll describe our results of that review as well to give some kind of context to where this is coming from. So, there we go. So the drivers behind an interest in AMR in plants in general, and the review that we undertook in particular, as we all know, the use of antibiotics in agriculture can drive the evolution of AMR. But I think it's fair to say that the vast majority of work done on this to date has focused on livestock agriculture, or the vast majority of work done in agriculture has focused on livestock agriculture rather than crop agriculture. But a couple of years ago, in the popular press, there were reports of increased antibiotic use and misuse in crop agriculture. So the picture on the right there is a picture of people injecting the trunk of a citrus tree with an antibiotic suspension in Thailand. There was also a widely publicised approval for the use of a couple of different antibiotics to treat, again, citrus trees in the US. These were both for one particular bacterial disease of citrus trees, citrus greening disease. So this prompted a kind of renewed interest in what the risk of AMR in crop agriculture was. So the review we undertook for DEFRA and FSA was really to kind of get a broad background of what diseases are being controlled by antibiotics around the world, how else antibiotics might be entering the crop environment as well, what other sources of resistance there might be, and really what are the knowledge gaps in our understanding of antibiotics in plant agriculture. So to undertake this review, we did an information gathering exercise comprised of two different parts. One was to try and elicit expert opinions from a range of experts around the world. So we contacted both our personal plant health contacts and also International Plant Protection Convention national named contact points spread across a range of countries around the world to try and get a broad geographical spread, as well as to try and focus on countries that might be of particular importance based on their exports of crop-based food products. And we also supplemented this expert opinion piece with a survey of the published and the grey literature as well. It's very fair to say that we got an extremely limited response to our inquiries from the experts. So this map here shows in orange all the countries that we contacted without a response, and in blue the very few countries that we actually got a response from. So we got a response from 6 countries in the end. Plus we did have a good conversation with the CRD, the Chemical Regulation Directorate in the UK as well, about the situation in the UK. So possibly interesting in itself that we got such a low degree of engagement potentially from plant health professionals around the world. But synthesizing those responses plus the results from the grey and the peer-reviewed literature, we got evidence that antibiotics are being used globally to control bacterial diseases of plants, in a number of diseases and a number of crop types as well. So probably no need to absorb all the details of this table. I'm sure you can look at it in the recording. But I would just point out as well that on the right there, streptocycline, obviously not an antibiotic itself, but refers to a 9-to-1 mixture of streptomycin and tetracycline that the grey literature reported or suggested was being widely used in India in high doses on a range of crops. Subsequent to this work that we did, there was a more recent paper by Chanvatik et al., and they produced evidence that even more antibiotics are being used in Thailand in mandarin production as well. So to summarise the status of direct application of antibiotics to crops, the primary use of antibiotics in plant agriculture that we were able to identify was to control bacterial diseases of pome fruits, so these fruits like apples, stone fruits and citrus fruits, plus vegetables, with the largest reported uses being to control fire blight, the disease of apples, and maybe now also citrus greening disease of citrus in the US. There is evidence that resistance to these antibiotics has arisen in plant pathogens and also in commensal organisms of plants. But the resistance mechanisms that we see in these organisms actually appear to be different to those that we commonly find in human and livestock pathogens. So there's a question there about, even if resistance is arising, how important is it from a human health point of view? Are these organisms or genes making it through into consumers? Some slightly more recent work as well by Taylor and Reeder, based on plant protection product recommendations in low and middle income countries, actually identified rice as being the crop that had the highest proportion of recommendations for antibiotic use, and also evidence of recommendations for clearly inappropriate use to control fungal diseases and invertebrate damage as well. So interesting there. So the kind of take home is that, I guess, where direct antibiotic use in crops is known, it is, it does appear to be quite a lot lower than in livestock agriculture, which is maybe not a surprise. But there are a lot of regions where regulations are weak or non-existent, and their use may be more widespread, and that's a bit of a knowledge gap, possibly. Possibly more important than direct application of antibiotics, certainly in European countries, I would say, really alternative sources in soil amendments. So things like manure, sewage, maybe grey water could be potentially important sources of antibiotics or antibiotic-resistant organisms in crop agriculture. Antibiotics can certainly be assimilated into plants, but what happens and where they go in plants varies a lot according to the compound itself, the plant, crop type, the soil type, and soil conditions as well. There's one other factor that might be potentially quite important from a crop health point of view in terms of antibiotics, and that is the phenomenon of co-selection. So where resistance to one group of compounds can also select for resistance to antibiotics. There are three major ways that this can happen. One is cross-resistance. So this is where the actual same mechanism provides resistance to both one chemical type and an antibiotic. So this could be, for example, the same efflux pump pumping out both a biocide or an antibiotic from a cell. There's actual classical co-selection. So that's where the genes to an antibiotic and another chemical could be located on the same genetic element. So that could be genes to an antibiotic resistance gene and a metal resistance gene being present on the same plasmid, moving around together. And the other one is adaptive resistance. So this is where, for example, exposure to a pesticide can allow a bacteria to tolerate higher levels of an antibiotic. And that could lead to stable resistance if the bacteria is able to survive these kind of high levels of an antibiotic and then separately evolve a resistance mechanism. From a crop health point of view, there is extensive evidence that metals can co-select for AMR, and probably most important from crop health is copper. So this is directly used as a fungicide in certain types of crop agriculture. Also could enter the crop environment through pig manure, could be used in pigs as a growth promoter, or from atmospheric deposition. There is quite a lot of evidence that some pesticides can lead to increased resistance to antibiotics, largely through adaptive resistance. I mentioned on the previous slide, but very few pesticides have even been assessed for their ability to do this. And maybe less importantly, there is also evidence that some organic pollutants and biocides can co-select for resistance or shift the microbiome to a more intrinsically resistant microbiome. And a few ideas for further work that fell out of the review that we did is that it would be great to have a better understanding of antibiotic biome accumulation in plants, because it's an understudied area. As I said just now, very few agrochemicals have been assessed for their ability to co-select for AMR. We got very few responses to our review, so maybe some methods, social science type methods to encourage participation in information sharing among plant professionals could be useful. And increased surveillance might help us to understand the actual risks of AMR in plants, because we know that resistance can evolve in the crop environment after application of antibiotics, but how frequent is this? Does this survive till it reaches the consumer or to the retail shelf? And are these genes actually making it into the consumer's microbiome or not? These are all knowledge gaps, I think. So I will wrap up there. Just say thanks to everyone who worked on the project. Very happy to take any questions now or in the chat or in the session later.
Great, thank you very much. Again, the microbiome coming up, this interesting thing that's coming through, but really quite terrifying how little we know about the importance of it, right? It sounds, those antibiotics that you listed are all ones that we recognise from the human health perspective. It's not a minor, they're not old or unknown antibiotics, so that's quite right. Okay, yeah, I'm sorry, but I think for the interest of time, I'll leave the chat. If people, there's one question that's come up to you, but it's not really a clarification one, so if you'd like to just answer that in the chat. And then now I would suggest that we have a 10-minute break, so to come back at 1:05, and then we'll hear the short perspectives from early to mid-career researchers. So another thank you very much to our first 4 speakers this morning. I think they've given us a really broad overview across the complexity of AMR across the One Health angles, and I hope that they can all remain for they're all available for the discussion session later. So yeah, so everybody, let's take 10 minutes now. Very welcome to stay connected to the Zoom workshop. Just leave your cameras and microphones off, and we'll see you back at what for me is 1:05, so in 10 minutes time. See you then. Okay, hello everybody. Let's make a start with the second part of our workshop. So the aim for this section was that we would bring together short perspectives from early to mid-career researchers. So these are all individuals that submitted an abstract outlining a key research gap or area that they felt needs further focus and development. So we're going to hear from 6 early to mid-career researchers in this section. So Finn, Nidhi, Yinka, Kuhn, Kevin, and then Fresco. And each of them has around 10 minutes to talk to that topic. So we're starting now at 1:05, so we'll have about an hour of these talks, and then we'll have hopefully a good at least 20 minutes for discussion at the end. So we'd like to start with Finn. Finn, if you'd like to share your screen. So Finn McQuaid is from the London School of Hygiene and Tropical Medicine, and he'll be discussing empirical treatment regimens for the programmatic management of drug resistance. Finn, over to you.
Fantastic. Thank you very much, Gwen. And so just to say from the outset that I am a TB researcher, so I'm very much focused on TB here, but I think some of this applies sort of much more broadly, and I hope there are things from it for others as well. So I want to imagine for a moment that you're in a clinic and a patient has presented with TB disease. You've diagnosed this and you know that drug resistance for TB can be a real problem. We have around 450,000 people getting drug-resistant TB every single year, and about 150,000 of those actually die of TB. Only around a third of people access appropriate treatment every year. So you know that drug-resistant TB can be a problem, but you might not have ready access to drug susceptibility testing. So you have to then make a decision on how you treat this patient. You could assume that they have susceptibility to your first-line drugs, in which case if they do have some resistance, then you may be providing ineffective treatment, which could also result in onward transmission of infection. Or you could assume that they have resistance to some first-line drugs and treat them accordingly, in which case you may be giving them treatment which is potentially toxic, more toxic, expensive, and there's also the possibility that they will then acquire resistance to some of these second-line TB drugs. Now, obviously, the risks vary by the individual in terms of whether they have risk factors, their age, their HIV status, and so on. But there are also population-level risks, and particularly when we're talking about empirical treatment, this depends very much on what the background level of prevalence of resistance is in the population, so whether they're likely or not to have that resistance or not. So the example that I want to give to try and think this through is of rifampicin-resistant TB. So rifampicin is one of the first first-line TB drugs, and we have relatively good drug susceptibility testing coverage for this in many settings. However, there's a key second-line set of drugs, fluoroquinolones, where these will often be used to treat people with rifampicin-resistant TB, and DST coverage for fluoroquinolones has increased recently, but it is still quite low. Only around 40% of patients receive fluoroquinolone drug susceptibility testing, and results for this can be really slow to come through as well. Now, the alternatives to this for people who do have fluoroquinolone-resistant TB treatment has historically included drugs such as linezolid, which can be quite toxic and can be quite expensive. So if we think through two different examples, in South Africa, there's relatively low levels of fluoroquinolone resistance. In Pakistan, there are much higher levels of fluoroquinolone resistance in rifampicin-resistant TB patients. In Pakistan, this is something like 50–60% of rifampicin-resistant TB cases also have resistance to fluoroquinolones. Now, in both of these instances, the National TB Programme had to make a decision about what treatment to recommend while waiting for drug susceptibility testing results, which in some cases, or in fact in many cases, never actually arrived. And in both instances, both national TB programs elected to recommend the use of linezolid until fluoroquinolone susceptibility was confirmed. And in Pakistan, this might make sense because quite a large proportion of people likely did have resistance to fluoroquinolones. But in South Africa, this might not actually make so much sense. But unfortunately, at the time, there was no real evidence, no quantitative evidence to support the people who are making this decision and developing these guidelines in that. So they decided that on balance, they would prefer not to risk people being undertreated and leading to onward transmission of rifampicin-resistant and fluoroquinolone-resistant TB. But they didn't have any quantitative evidence to support them in making this decision. So we wanted to think about how we might improve the evidence basis here for them, or think about how to help them make this decision. So we imagine we have a hypothetical cohort of individuals. They have resistance to rifampicin, and they have some unknown level of fluoroquinolone resistance prevalence. Let's for now concentrate only on those where we don't have second-line drug susceptibility testing, and think about two regimens. So we have one regimen over here, which is a regimen which is intended for people with fluoroquinolone susceptible disease. So it's relatively successful at treating them, but not very successful at treating people with fluoroquinolone resistant disease. Then we have a second regimen, which has intended for people with fluoroquinolone resistant disease, and it has relatively high levels of treatment success, so sort of low deaths in this instance, irrespective of the resistance status over here. However, this comes at a cost in terms of an increase in serious adverse events. So I've just chosen these two indicators, deaths and serious adverse events here, but really it's a problem that is more sort of generally applicable. So here we could be thinking about treatment success. Here we're just thinking of the sort of the things like cost, monetary cost, serious adverse events, the potential increase in acquisition of resistance, and so on. So we could be looking at any sort of indicators here, which kind of indicate the cost of using an inappropriate treatment or of over-treating here. So if we have these two different regimens, and in TB, one of the issues we're having with, or it's a good thing, but it is still representing some challenges, is that there are a large number of treatment trials ongoing at the moment. So in fact, this example would have been more appropriate 6 months ago, because we now have regimens which do treat fluoroquinolone resistance without that sort of increased toxicity. But there are a lot of different regimens coming out, and it's quite difficult for people to make these decisions on how to do this empirical treatment in this kind of changing environment. So imagine we had these two regimens, that we had one for fluoroquinolone susceptible disease and one for fluoroquinolone resistant disease. And we wanted to decide which regimen to use. Well, if we knew something about the background prevalence of resistance, then we know that as the prevalence of resistance increased, then using a regimen that's intended for people with susceptible disease would come at an increased sort of risk of treatment failure and death and so on. And that wouldn't really be the case for the drug, for the regimen intended for people with resistant disease. However, that would come at a cost in terms of adverse events or dollars or whatever that happens to be. And here that would be higher for the second regimen that's intended for people with resistant disease. Now we want to ask the question, if you had one regimen, let's say the red regimen here, when should you switch to the blue regimen? So you can imagine that if you're in a country that has, or some setting that has a sort of interim level of prevalence of resistance, there's some benefit to switching from the red to the blue disease. You decrease mortality, but that does come at a cost in terms of increased adverse events occurring. And so that might be somewhere like South Africa. But if we have higher levels of prevalence of resistance, you can see that there's a much greater increase in benefit to switching and a sort of similar level of cost to switching. We can represent this in terms of the sort of the adverse events that would be incurred for every death that we averted. And I said before, we could be thinking about other indicators here, things like cost effectiveness, so thinking about dollars per daily averted and so on would be, it's a very sort of, it's the same sort of way of thinking. But how does that change depending on the background prevalence of resistance? So in the first instance, when we've got very low prevalence of resistance, there's really no benefit to switching from the red to the blue regimen. So we end up saying we should always stick with the blue regimen. But then we have to make the decision, what is our sort of willingness to pay threshold? How many adverse events are we prepared to accept per death averted? Would you be prepared to accept one adverse event? Maybe what about 10? What about 100? Wherever you make that, wherever that threshold lies, we then know that to the where that intersects here, to the left of that, when we have lower levels of prevalence of resistance, we should use the red regimen, and then when we have higher levels of prevalence of resistance, we should switch to the blue regimen. So you can see that you can make a very different decision on which regimen to use depending on the prevalence of resistance in your population. But there's a lot more to it than that. I mean, there I used adverse events. I think one of the really interesting things to think about here would be amplification of resistance, kind of comparing those, the long-term benefit of not developing more resistance to second-line treatment compared to the short-term benefit of actually successfully treating people. There's, as I mentioned, the sort of changing resistance profiles and TB regimens at the moment. And there are a lot of questions to think through around costs. And one of these, which I think was really interesting, given what Caroline said at the beginning, was what this tells us about the value of drug susceptibility testing and resistance surveys. And you mentioned at the beginning that there's been a sort of lack of funding in terms of diagnostics and surveillance, which I think was really interesting here. And then in the interest of time, so obviously there's also the issue of what matters to different stakeholders in this situation. But I think that in the TB world, at least,
wrapping up there.
Yeah. In the TB world, at least, I think this is a really big issue. There are a really large number of people who are being treated empirically, and we have to accept the reality that DST coverage is not widespread enough or quick enough. Obviously, we should be advocating for that to be improved, but in the meantime, national TB programs have to make this decision about how do they treat people. And I think we could be doing a lot more to provide them with quantitative evidence to make this decision-making better. Thank you.
Thanks, Finn. And I think what you said at the beginning was right, in that this is an issue not just for TB, but across many different other bacteria. Leonid has some questions about coverage of DST versus resistance that you can answer in the chat, I think, Finn. Okay, in the interest of time, let's move on. Thank you very much. Let's move on to Ninti Dadija from Imperial College London. Apologies if I pronounced that wrong. They're going to discuss the role of infection prevention and control in tackling antimicrobial resistance. So, hello, we can see you and we can see your slides. Can't hear you yet.
Can you hear me?
Yes, I can hear you now. Great. Okay. I'll switch my camera back on at 9 minutes. Okay. So when I pop back up. No.
Yeah,
right.
sure. Thank you so much. Hi, everyone. My name is Nidhi, and I'm a PhD candidate at Imperial College London. I'm in the NIHR HPRU in Healthcare Associated Infections and AMR, and my PhD is funded through the MRF National PhD Training Program in AMR research. So I'm here to make a case for strengthening research focus and funding for infection prevention and control implementation research. So I'll start off by just sort of reaffirming what we all know, that IPC, it's been well established that IPC is critical to tackle AMR. This has been highlighted in numerous ways, and one of the sort of high profile ways was in the UK AMR review, which highlighted the role of sanitation and hygiene in preventing infections. And, you know, at the global level, the WHO has included infection prevention and control in the AMR Global Action Plan as one of the 5 strategic objectives, you know, in reducing the incidence of infection. So the challenge of AMR is very closely linked with hospital-acquired infections, and we know that effective IPC reduces the spread of all types of infections, including resistant ones. But from what we see from studies, the situation is quite shocking. Up to 90% of health workers do not adhere to recommended hand hygiene practices due to many factors, including the lack of an enabling environment, On average, 15% of patients in low and middle income countries will acquire at least one infection in acute care hospitals. And globally, drug-resistant infections are killing more people than susceptible infections. And hospital-born babies in low-income settings are at a higher risk of being affected by neonatal sepsis, with infection rates 3 to 20 times higher than in high-income settings. Conditions in low and middle income countries are creating what we're referring to as a perfect storm for hospital-acquired infections. There are more complex care services being offered. Patients are admitted for longer. We have more hospital births for babies. But the IPC capacity and infrastructure is not keeping pace. Now, we have a lot of information and guidance on what to do for strengthening IPC. This is just an example of what the WHO has on offer for countries and facilities. We've got the guidelines on core components of IPC at national and acute healthcare facility levels. This is an excellent document that lays out across the health system, all the things that have to be done. We've got the WHO implementation handbook for national action plans on AMR. So we have all the knowledge and the information, but something is going wrong. In the WHO's first ever global report on IPC, which was released this year, it was reported that only 16% of healthcare facilities met all the WHO IPC minimum requirements. None of these were in low-income countries. And even in high-income countries, 27% of primary and 11% of secondary and tertiary facilities were meeting these requirements. In the TRACK survey, the global database for AMR country self-assessments survey, 33% of countries have no national IPC program or they're not being implemented. And only 35% of IPC programs are being properly implemented in healthcare facilities nationwide and being monitored. So there's clearly a gap between what we know and what we're doing, the no-do gap. And one of the ways that research can help address this is through implementation research. And what I mean when I say implementation research, we look at the Latin word implere, to fill up or to fulfill. It's the scientific inquiry into questions concerning implementation, the act of carrying an intention into effect, which can be policies, programs, or individual practices. And this form of research addresses implementation bottlenecks. It identifies optimal approaches for a particular setting and promotes the uptake of research evidence. We know that low and middle income countries face a substantial challenge, and we don't really understand them with IPC implementation. Yet there's a significant dearth of implementation research in the AMR world. My colleague Esmita Charani has conducted a review of evidence and stakeholder perspectives on research priorities for AMR. And what we see in this figure is it shows the current global funding bodies active in AMR research for humans. And you can see the broad themes under which funding is available. Implementation is that small purple slice. So this study really highlighted the imbalance in funding. We have a great emphasis on new drug R&D, but without really sort of filling this no-do gap and promoting and supporting more research, we're missing a trick. Some of the IPC research priorities for hospital-acquired infections and AMR include that we have a weak evidence base behind what is considered many of the best practice IPC recommendations, particularly for low middle income country contexts. We don't really fully understand the factors that facilitate success, as well as the barriers and challenges to implementing effective IPC programs. A lot of the current IPC research and recommendations focus on healthcare worker adherence to guidelines and training, but we don't have a good understanding of the implementation context and systems influences. And as was highlighted earlier in the workshop, we have a very limited understanding of the budget impacts of IPC measures for sustained implementation. So through my PhD, I'm trying to address some of these aspects by looking at strengthening IPC for hospitalized neonates in Botswana, which is my home country. And I'm using a systems approach to identify the system level factors for influencing IPC in hospitalized neonates in a tertiary hospital in Gaborone, Botswana. Botswana, in this hospital in particular and across the country, they've experienced a number of outbreaks within the neonatal unit that has resulted in significant morbidity and mortality. And neonates, as I mentioned before, are particularly vulnerable patient group. I'll be estimating the budget impact of current and priority IPC measures to help decision makers. And I hope to be able to identify leverage points within the neonatal unit and organization of care that can help inform stronger design and implementation of IPC to help prevent and reduce hospital-acquired infections. Finally, I just wanted to highlight that the COVID-19 pandemic has really revealed the urgency of IPC as a global priority. COVID-19 has exacerbated existing vulnerabilities within healthcare systems. And just this past month, the 75th World Health Assembly approved a resolution for a global strategy on IPC, with IPC as central to health emergency preparedness and response. And they highlighted that it was key to addressing the silent burden of hospital-acquired infections and AMR. And they positioned IPC in the context of health system strengthening and high-quality care delivery. So I think this is a really positive step, and I really hope that the momentum from this can be used to raise sort of funding and focus on research for IPC and in the implementation research sphere. So thank you very much, and please be in touch if you have any questions, or it would be great to connect if there are opportunities to collaborate.
Great, thank you very much. Thank you very much, indeed. That was a really nice presentation, and really important that you emphasize yes, what we've learned from COVID-19 and how this is really becoming central stage now, preventing infections instead of just treating them better, right? This is really what we need to be doing. I don't think any questions have come up in the chat yet, but if you have any questions, then do, and we can have more discussion later about IPC versus effective antibiotic usage. Thank you very much. Okay, for our next talk, we're going to be hearing from Yinka Samorin from the National University of Ireland, and they'll be discussing tackling AMR in the human health context. Welcome. I can see you, but I can't hear, and I can see your slides.
Thank you very much, Gwen. So I'll be giving a brief perspective on the gaps in antimicrobial resistant research, and I'll first start by thanking Caroline for the invitation to present today. So my interest in antimicrobial resistance started with some work I did during my PhD at the Queen's University in Belfast, where I was looking at antimicrobial resistance in urinary tract infection. Now, UTI is a common infection in primary care in the UK, and it's one of the bases for the most prescription of antibiotics. So the work that we did then looked at antimicrobial resistance in urinary pathogens in Northern Ireland. And then based on that understanding and the fact that prior to that time, prior to a few years ago, trimethoprim would be one of the major first-line drugs for treating UTIs for the empirical treatment. And there is quite a huge resistant problem to trimethoprim. So there was the need to change that prescription, change prescription, which basically then turned to nitrofurantoin. But there are some cohorts, some people who would not be able to get nitrofurantoin for the treatment of UTIs. So based on that, there's still the need to be able to rapidly eliminate or to make a decision on those that will still have that trimethoprim treatment would still be useful for. So We developed a multiplex assay for determining a range of organisms that could cause urinary tract infection, as well as trimethoprim resistant genes. And we just published some work on that, as well as, so because that project was done with a clinical partner, that assay has now been translated into one of their testing platforms, and there would be, it's undergoing verification at the moment. But beyond this, it's very clear that antimicrobial resistance is it's a big problem. And there was a recent review that looked at the global burden of antimicrobial resistance, showing that almost 5 million deaths associated with antimicrobial resistance, and specifically about 1.27 million people would have died in 2019 as a result of antimicrobial resistance. So the highest burden of that is in sub-Saharan Africa, and specifically in West Africa, which is the region I am from originally. I was raised in Nigeria. So because of this huge problem, it's antimicrobial resistance, and most of the deaths have been attributed to lower respiratory and thorax infections, bloodstream infections, and abdominal infections. UTI is the fourth of those. So in the course of the perspective I'll be sharing basically would be on some of the factors that make antimicrobial resistance a big problem in West Africa, and maybe end with some thoughts on how that effort could be put into that. So first would be urbanization. So there's been evidence of the fact that with increasing population, there is the higher risk of antimicrobial resistant transmission, especially based on the impact on the infrastructure deficit that are in this peri-urban environment, as well as access to antimicrobial to antibiotics. So it's then very instructive to know that because of the dense population, the hygienic conditions of the people, it's definitely much reduced. And the fact also that in these kind of contexts, rearing of animals, poultry, and the fact that the human environment and the animal condition is such that there's such a close proximity, these could increase the interaction and the exchange of mobile genetic elements, which definitely would make there to be an increase in the transmission of antimicrobial resistance. There's also the problem of food production. So within food production, antimicrobial resistance has been identified as a problem. So for example, there've been antimicrobial resistant pathogens observed in the production of several food products, especially, well, the more concerning ones would be the ready-to-eat foods, which usually would not undergo any further treatment before they are consumed, and that could lead to transmission directly from these foods to humans. Also, the environment where these foods are traded or agri-produce are traded could be such a hub for the exchange of antimicrobial resistant pathogens. So my perspective on the reduction or tackling antimicrobial resistance in this context would be on two main things. And the first would be the importance of reducing the need for antimicrobials. So with these interventions that need to happen would always have to be within the local context, because without understanding the drivers of antimicrobial resistance in a particular environment, it's much difficult to address it. So it's important to understand the burden of AMR in local context. And I would describe that with two potential ways we could address that. First would be to increase surveillance. Now, there've been effort at doing this already. So for example, the Fleming Fund they gave a grant to Nigeria, and they built infrastructure for increasing surveillance. However, most of these are top-bottom approaches, which tend not to get to the primary care level or even to the communities where most of these transmission goes on. So it's important that we do that. And one of the things I've been doing in this area would be, I've been working with the local hospital in Nigeria to look at what the local context of their antimicrobial resistance, following up from the work I did at Belfast, to look at how to understand the local context of UTI and what are the resistant problems that's associated with the treatment, so that we could come up with potential rapid diagnostics that would help. Secondly, in understanding the local context would be the importance of improving infrastructure, and not just infrastructure, but the people to drive that infrastructure. So many labs, especially at the primary care, would not have facility for actual diagnosis, which is identifying pathogens and doing susceptibility testing before they prescribe. So I'll talk more about that in the next slide. A very important factor that also contributes to infection would be the sanitation and the hygiene. So it's important while looking at addressing antimicrobial resistant problem within not just the human health, but the environmental as well as the animal health perspective, that sanitation is addressed, which would help reduce infection. Also, another point there would be vaccination. So it's important that research continue to develop vaccines towards some of these pathogens that carry antimicrobial resistance markers. The second major theme would be the reduction for minimizing the need to use antibiotics. And I would focus here on the prescription practices. So most of the, there was a study that showed that in Nigeria, only about a quarter of patients that were prescribed antibiotics really had a thorough diagnosis that they were from bacterial origin, which means that just treating by symptoms only would often lead to misuse, overuse of antibiotics. Also, in health workers that prescribe, so it's not only medical doctors or pharmacists that prescribe in Nigeria, at the primary care level in many local communities, the community health workers are also able to prescribe antibiotics, and without proper diagnostics or proper identification of what's the real pathogens that are causing this antimicrobial resistance are involved, it's difficult to treat effectively. And then lastly is that because of the access to antibiotics, some people would just randomly use antibiotics once they're having a symptom, which would be inappropriate. So in addressing some of this, I would propose that for the symptom-based diagnosis, to overcome that, there is the need to develop rapid diagnostics, and we already are working in that area, which there is definitely need for a global effort to address that. It's important that there is also education of not just the healthcare workers that prescribe, but also the people, the population, so that they would understand by this education, they would be able to have behavioral changes which will then reduce or minimize the use of antibiotics when it's not necessary. So this is just my brief overview of the work I've been doing around antimicrobial resistance and some of the gaps we think that needs to be addressed. And I'm happy to have questions if there is any. Thank you very much.
Thank you. That was a really great overview of, as you say, the importance of context and understanding what's happening and the gaps needed. Thank you very much. In the interest of time, I think you've had a question come up in the chat if you want to answer that, Inka. Thank you very much. We'll now move on to Kuhn. Are you there, Kuhn? He's going to be discussing, antibiotic prescribing strategies for common conditions in companion animals and the consequences in terms of AMR. So we're jumping from a West African UTI drivers to a UK, possibly UK, I don't know, companion animals focus. So passing over to you now, Kuhn from the University of Oxford.
Yeah, thanks. It's not all UK, so it's a bit EU focused, this particular talk. So basically, it's about something that has been highlighted before as well, and focusing especially on what actually is the influence of prescribing decisions on the microbiome. So something that was made me quite happy, but also sad, because it means there will be a lot of more pressure on some of the projects I'm working on, is the recent G7 meeting in May 2020, where there was a lot of focus and commitment to contribute to establishing new and improving existing national surveillance systems on AMR, and also a particular focus on reducing antibiotic use and monitoring it, both in volume and appropriateness, with a special focus on usage in humans. Unfortunately, in that paragraph, there was no mention of use in animals. And while most of my work actually is focusing on use in animal and humans, I also started recently working a bit with veterinarians to address the issue of potential overuse in that sector. So to give a bit of context, I first had a look at, well, has anything worked so far in terms of reducing unnecessary prescribing? Well, there's some really nice examples, for example, in the Netherlands, where there was more than a 50% reduction, safe reduction in antibiotic use for livestock between 2007 and 2012, without subsequent negative consequences. But to put that in context, that happened after a doubling of approximately similar increase in the years before that. So maybe we're just back to where we were, and that might not be good enough. Who knows? And in terms of antibiotic use in English primary care, where I've done a lot of my research, is well, in recent years we have a nice reduction, but again, before that it was actually a substantial increase in daily defined doses per inhibitant, and ironically that increase started roughly when we had the first national action plan implemented in 2000. And as many of you are aware, probably the most effective intervention not made by humans in reducing prescribing was COVID, where during COVID, prescribing in primary care went massively down. So in terms of what about companion animals? Well, because we saw huge reductions in food-producing animals in the UK, which is good, but we didn't really see similar declines among companion animals. And there's a lot of broad spectrum and critically important antibiotics that are being used in companion animals in Europe, as a large survey has shown. And lowering antibiotic use, or at least unnecessary antibiotic use in companion animals, is important for several reasons, including, of course, lowering the risk of side effects for the treated animal, potential development and transmission of AMR within the treated pet, but also potentially onward transmission of the resistant bacteria themselves, or transfer of mobile genetic elements to owners and other household members. And of course, a frequent close contact between companion animals and their owners provides opportunities for bidirectional transmission, as also assessed in the JPIAMR pet risk program. But what is really unknown, at least to me and to most of my colleagues, is what the impact of reductions in antibiotic use has on antibiotic resistance, and particularly among bacteria that are also part of the microbiome at the time of antibiotic treatment, as already highlighted in a few talks. So we really need more evidence on, basically, if we think about what antibiotic treatment decisions are being based on and what they need to balance, they first need to balance the clinical benefit of antibiotics versus the selection and development of antibiotic resistance. And while we know the first part relatively well from several trials, the latter part is largely unknown. And actually, if you ask clinicians and veterinarians, they often hugely overstate the clinical benefits. So although there are trials, if you ask them about the number needed to treat, they substantially underestimate the number needed to treat. So they think, oh, I only need to treat a few patients to get better, to save one event. But in practice, that is often much larger for common conditions encountered in primary care. So the safety and impact of not prescribing an antibiotic for common conditions would ideally be assessed more properly and quantified using non-inferiority trials, testing either behavioral interventions or just directly no prescribing strategies. And something that's close to my heart and which I started doing a bit more is trying to also find optimal durations of antibiotic treatment, again using non-inferiority trials estimating optimal duration. And in terms of the latter, for many infections, the optimal antibiotic treatment duration is unclear. It's often based on some arbitrary numbers that we prefer, but it should be long enough to treat infections effectively. But at the same time, you want it short enough to not influence the microbiome too much. But we don't know really what is most optimal treatment duration. And while this evidence is relatively weak in humans, in companion animals, there's virtually no evidence at all. There are some very small studies for UTI, but as far as I know, there's not much out there otherwise. And just as an example, which I often use, so we see here this lady that in humans we recommend 3 days of nitrofurantoin for most people with uncomplicated cystitis. And this little friend in the corner, in many European countries, is actually recommended 7 to 10 days of often co-amoxiclav, which seems to be the overkill for this little friend. So how do we traditionally assess this in humans? We often do a head-to-head comparison of 2 arms. But the problem is if we do a non-inferiority trial, and we find that the shorter duration is worse than the longer duration, we only have learned that, and we don't know anything about this part. So it might well be that 13 days is inferior, but that, let's say 16 days is not inferior to the standard duration. So we could make substantial changes, but what that means, we don't know yet. So the solution is really to randomize patients to multiple different durations and estimate duration response curve. And some simulation studies from Matteo Cartegno have nicely shown that you only need about 500 patients for lots of these questions. And then you can find the shortest duration that's non-inferior to standard duration. So what could funding really add, and what is really missing? Well, so far what we already have done with a group of veterinarians and academics across Europe is designed, because if you do a non-inferiority trial, you really need to pick your non-inferiority margin wisely. So what difference between the outcome are you potentially willing to accept, where you can't say, well, the difference between the treatment is not larger than X amount. And often this is done just by the researchers, but ideally you would have something that's acceptable to actual practicing veterinarians. So that's something we have designed with surveys to elicit. We have simulations in place to estimate the sample size required for antibiotic non-inferiority trials for several conditions. But I think what ideally we would have is funding to establish such platform trials and bring in together networks around Europe that can make such trials happen, and not only directly assess the clinical outcomes that we often know a bit more about, but also focus on the other side of the coin. So focus on the impact of antibiotics on the gut microbiome, both in patients and their household members. And importantly, because of what a lot of clinicians often tell us, is that, well, even if you find changes in the microbiome, I don't know what that means. So we really need to assess what do these consequences of changes in the gut microbiome mean on subsequent clinical outcomes. So having longitudinal follow-up on clinical outcomes, and I'll end it there.
Thank you very much. Well, got microbiome coming up again.
All right.
No, that's great. So that's really interesting that you actually have numbers for how many clinical trials patients you need for a non-inferiority trial. But great. Okay, so let's move from companion animals, thank you very much, Dean, to humanitarian crises. So we're going to go back to the London School of Hygiene and Tropical Medicine now to hear from Kevin. Kevin van Zandvoort is going to talk about the burden of AMR in populations affected by humanitarian crises. So I can see your slides, I can see you.
Great, can you hear me as well?
Okay, yeah, I can hear you too.
Brilliant.
Okay,
Thanks, Gwen.
see you in 10 minutes.
Hi everyone, I'm Kevin. I'm an epidemiologist here at the London School of Hygiene and Tropical Medicine. I'm going to talk about the AMR burden in populations that are affected by humanitarian crises. First of all, what is a humanitarian crisis? Essentially, it's any event that threatens the health, safety, or well-being of a large group of people. And crises can be caused by war, natural disasters, famine, and disease outbreaks. Actually, we often only think about war, but there's a number of causes of crisis. Currently, there's several ongoing. So there's a few very high-profile ones in the acute phase, including the war in Ukraine, the hunger crisis in East Africa, and the civil war in Yemen, and several protective phase crises as well, such as the conflict in northeast Nigeria, the long-standing instability and conflict in DRC, and the, for instance, the hyperinflation and malnutrition in Venezuela. But of course, there's many others. If you look at the right-hand side of the plot, this essentially shows the number of displaced people worldwide, estimated by UNHCR. And yeah, just to show you the shockingly high number of over 80 million people who are forcibly displaced worldwide. So that's essentially 1 in every 100 people worldwide. So it's a very large population we're talking about. And a population that has been increasing over the last 10 years, from about 40 million to 80 million now, and still increasing. When we look at the health burden of humanitarian crisis, first of all, it's very complex and it depends on the situation, and no two crises are the same. But essentially what we do see in most crises is an increased disease and mortality rates, where all-cause mortality is often at least 2 times that of the pre-crisis baseline. Respiratory tract infections and viral diseases are the leading causes of mortality, and we don't really know the etiology of those infections, but it's very likely that at least a substantial proportion of those are caused by bacterial pathogens. Also, we know that in many crises, there's a high presence and prevalence of several risk factors that can aid the spread of infectious diseases that can increase both transmission, but also the risk of infection actually leading to severe disease, death. These include acute malnutrition, overcrowding, and altered social contact patterns, disrupted routine vaccine use, low access to curative care, inadequate water and sanitation and hygiene, and of course, as well. When we look at the AMR burden in humanitarian crisis, it's really largely unknown. And that, of course, results in suboptimal treatment and care for the population. It also hinders the development of effective mitigation strategies. For instance, recently I was involved in, we are actually applying for a study, funding for a study together with MSF and Save the Children to evaluate the impact of a mass drug administration of azithromycin in children under the age of 5 in acute phase crisis. And we don't really understand the pre-existing burden of AMR in at least the average burden in most of these settings, which really makes it difficult to understand the potential impact that such campaign could have, or even the potential increased AMR risk of doing such a campaign. Even though we don't really understand the burden, there are, again, as well, several risk factors present in those crises that can both accelerate the emergence and spread of AMR. Crises, unfortunately, often happen in settings that already have weak health systems and a high AMR burden before the onset of the crisis. There's often broken or unreliable supply chains for drugs, which can result in a rise in the use of substandard or expired drugs, but also self-medication with suboptimal therapies or inadequate dosing, which can, of course, result in emergence of AMR. Also in war zones, what you often see is the environment gets contaminated by heavy metals from the use of munitions, destruction of buildings, again, causing selection pressure, potentially for AMR development. And there's the list of risk factors that I mentioned before that can result in increased transmission of pathogens and therefore also resistant pathogens. There is a bit of available data in the settings, and essentially wherever they found high resistance rates in most crises. For instance, in the DRC, they found that 75% of bacteria in neonatal sepsis cases were resistant to recommended antibiotics. In Yemen, Syria, and Iraq, they found high rates of chronic bone infections caused by drug-resistant organisms in war-wounded patients. In Niger, high resistance in severely malnourished children, and in Libya, high rates of TB, probably MDR TB, in overcrowded detention centers. In addition, when we look at antibiotic prescription or consumption as kind of a proxy, there's also little data available on those. There's no routine surveillance in place in most settings, and also often little capacity to do some research for storage or analysis of samples. However, there have been some developments. So there's research projects looking at or collecting samples that can be used to look at AMR in populations. So myself, I'm involved in a project where we like native swaps and internal people, which we assess for pneumococci. MSF has used blood cultures to look at the etiology of sepsis in children. And some places point prevalent service that we need to understand at least antibiotic prescription patterns on a single day to get some idea of what kind of antibiotics have to work. And prescriptions are quite high. In addition, MSF has developed this called NIDAP, which is essentially a standardized solution for microbiology in low resource settings that they can out to settings. But essentially, it's not really enough to fill this knowledge gap and improve the access to this population. So yeah, there's a very clear research gap, and more research really needed to understand what is the true AMR burden to address these issues. Of course, one reason for that is that research in these settings is notoriously quite difficult. In very resource-constrained settings, the populations are often transient, and people they may move in or out areas like IDP camps. Often you have only intermittent access to the population. There might be safety concerns, for instance, if we're talking about a war zone. And of course, research not the priority of humanitarian actors when they're doing their humanitarian response. So there's really a space here for research to be done and research to be funded. And as we've seen, that there's a very large amount of people that is affected by these crises, but not really any plan to address their specific needs in regards to AMR, but also many other issues. Then prioritizing AMR research, or really consider much in the development of, for instance, diagnostic tools to get drug susceptibility. And yeah, generally often overlooked, but they really should be included if you want to look at a truly global AMR response. So yeah, thank you very much. And if there are any questions, I'm happy to take them.
Thanks, Kevin. That was really great and really interesting, as you say, to raise the fact that this is not a small minority problem, right? This is a huge number of individuals. And this, I'm trying to think whether this is the story to sort of say, is it the problem that people are getting infected with drug-resistant pathogens in there, or when they leave the camps, or they leave wherever crisis, or once the crisis is resolved, then in the future they're then colonized asymptomatically, and that's what they go on, or we just don't know yet, basically.
I think we don't know, but I think probably both, right? Like in these camps, you have a huge, just a lot of amount of people like coming together in one place. So you have a lot of overcrowding, a lot of transmission going on of infections in general, and resistant pathogens as well if they're introduced. And then there's all these other reasons that might risk the development and emergence of AMR in these settings. Yeah. Again, can spread, and potentially then, spread in host populations as well. So yeah, they're just spreading out of just the crisis affected populations. Yeah.
Great. Well, not great, but great that you raised it here. Thank you very much.
Cheers.
Great. Okay, we'll move to our final speaker for this early and mid-career focus session. So it's my pleasure to welcome Tresco, another London School of Hygiene and Tropical Medicine researcher. They will be discussing AMR research to inform national policy questions. So over to you, Tresco. Thank you.
Hey, can everyone hear me all right?
I can hear you and see you. I can't see slides yet. There we go.
What about now?
Yeah, I can see slides.
All right, let's do this. Presentation is quite long, but I'm not going to actually read the whole thing, just in interest of time. So basically, this presentation is about AMR research, specifically focused on antimicrobial use in agriculture to feasibly inform national policy decisions. So you can skip that. Basically, when we want to inform policy analysis relating to antimicrobial use in agriculture or any other AMR issue, we want to understand, what effect do these policies have on the amount of AMR infections in humans, and also what is the full societal effect of this change in the incidence of resistant infections, and what is the effect of this policy on other aspects of society, on the economy, on agriculture, and that sort of thing. And a lot of research in this area doesn't really touch on these. They may look at the relationship between bacterial communities in humans and animals from a microbiological perspective. They may consider the impact of this, but it tends to focus on human health as if AMR is just a human health issue. They may say how many incidents of disease, how many deaths, how many qualities lost even. But this does not really give us all the information that we need to make policy decisions. When the economic methodology is applied, it may be very simplistic. It may make statements like, oh, we assume that if antimicrobial use society-wide goes down by 10%, then the amount of resistant infections in humans goes down 10%. When in reality, there's no statistical or theoretical basis for that. It's just a convenient assumption to make. And this is the assumption made by the recent OECD modeling study. So we kind of need a better idea of how to inform policies. And there have been some really fantastic comprehensive frameworks like Chantal's recent paper, helping us understand the full impact of AMR through all the different pathways. And basically, we know the different ways in which AMR can affect society. So how do we take these understandings and integrate them into a model to inform policy in a way which can easily be done with existing data? So for example, let's say government wants to ban the use of antimicrobial growth promoters, but knows there may be a trade-off with agricultural productivity. How do we weigh this effect on agriculture against the effect on human health? What if we reduce antimicrobials but also improve WASH? And how is this going to affect the amount of antimicrobial resistant infections in humans? Won't this affect the economy? And won't this also have knock-on effects like using up healthcare resources? And what about surgical site infections in a world with more antimicrobial resistance, et cetera, et cetera, et cetera? It's a lot, and it's difficult to answer all of these questions, especially mechanistically, and especially in situations where there aren't very good data. So basically, here is a simplified framework based on an earlier paper that is still in preprint for how we can analyze this using statistical and mathematical models. So let's say we change the amount of antimicrobial use in farms, and this will have some effect on farm productivity, which we can measure. And this may also be mediated by the characteristics of the farms and other interventions. And then through the effect on food security, this can also affect morbidity and mortality in the population, which we can attempt to measure. This change in the amount of antimicrobial usage will, as far as we're aware, affect the amount of AMR infections in humans. And this can also be mediated by country characteristics. And then indirectly, by using up healthcare resources, affecting surgical site infections, and so forth, this can affect indirect morbidity and mortality that is not directly from antimicrobial resistant infections. And it can also affect the amount of morbidity and mortality from AMR infections. And this in turn can affect the economy through labor productivity losses. So essentially, if we look at these outcomes, so the direct morbidity and mortality, the effect on labor productivity, the effect on farm incomes, and the indirect effect on all-cause mortality via strained healthcare resources, food security, and so forth, we can get not entirely comprehensive, but still very comprehensive idea of the societal impact of changes to AMU in agriculture and other AMR policies, which can be estimated using statistical and mathematical models. So how will we approach this? So what I'm proposing is a very condensed framework, like the minimum that you would need to be able to answer this question comprehensively. Essentially, it has a set of statistical models, which then feed into a mathematical model. And these models would be estimating the relationship between AMU on farms and farm output, either through large-scale farm trials or where we have good data sets on farms using those, estimating the relationship between farm output and all-cause morbidity and mortality, controlling, not via the relationship between farm output and population level morbidity and mortality through food security. And for this, we could use an instrumental variable technique like using weather shocks. And then estimating the relationship between how much antimicrobials are used in animals and the incidence of resistant infections in humans. And for this, we could use panel data technique at the population level. And then finally, the effect of AMR infections on all-cause morbidity mortality, not through the infections themselves. So, for example, through healthcare resources being strained, through surgical site infections and stuff like that. Basically, by regressing all-cause morbidity and mortality on the amount of antimicrobials used in agriculture and controlling for the amount of AMR infections, as well as all other relevant factors. This can then be fed into a population level mathematical model using Markov state transitions with humans and farms, and can be parameterized to the relevant country. And the neat thing about this is we can actually use the statistical models from part one to parameterize this as well, to understand the intervention impact both on human health, the wider economy, and agriculture, indirectly and directly. So we can attribute quality adjusted life years to all of the morbidity and mortality caused by direct and indirect effects of antimicrobial use. Using a willingness to pay threshold, we can assign a monetary value to this. We already have a monetary value assigned to the effect on labor productivity and farm outputs. And we can essentially compare these outcomes in like terms, because policymakers do not only care about human health, they also care about the interests of all stakeholders. And by comparing these in like terms in this way, we can understand the full societal impact of AMR policies. For this example, I've talked about antimicrobial use in agriculture, but it could really be anything. And this may sound overly ambitious, but it's not too dissimilar from a paper that we submitted a few months ago, which is still in preprint. The only difference is that for that paper, we were not able to parameterize all of these statistical relationships. So we had to basically use the existing literature and come up with a set of feasible values for all of these statistical relationships. But using the methods that are being suggested in this presentation, we can estimate these relationships specifically for whichever country we're looking at. And this will allow us to actually understand how antimicrobial use, antimicrobial resistance, human health, and the economy are related to each other in real terms that can be fed into policy decision-making models. So, as I said, this example focuses on antimicrobial use in livestock, but it doesn't have to be just focused on that. Instead of looking at farms, we could look at the environment or other human health components. We could look at the effect on pharmaceutical revenues. If, for example, we were looking at a regulation on pharmaceuticals, we could look at the environment. How does this affect the replenishment or depletion of natural wealth? We could look at for example, if we're looking at a policy which restricts the use of antimicrobials in humans, we can also look at how this might affect the prevalence of susceptible infections and weigh these against each other. The beautiful thing about this is that it allows us to compare disparate effects in like terms. And I think that for policy decision making, especially at the national level, which is where these policies are ultimately made, we have to be able to compare all of these in like terms. A cost effectiveness analysis is best when it includes the effect on the economy, on human health, and on all relevant stakeholders. So just in the interest of time, I'm going to stop there. And I understand this is very rushed, but this is why there's a lot more text on the slides. So if the slides are available afterwards, as I think they are, I really encourage you to look over them in more detail. And maybe you have some better ideas. I'm open to hearing them. And if you thought this framework was neat, please use it or improve it or think about it or do whatever you want. This kind of framework, I hope, can guide future research. So I encourage you all to use it for that purpose. Thank you.
Thank you, Chesko. That was a great overview of this framework. And in some ways, you neatly wrap up with what Chantal started with, with this need to understand what policymakers need, which is an economic evaluation of priority setting to inform policy decisions. And how can we do that? By integrating data across the different environments or fields or perspectives, right? Really interesting. Thank you. Okay, right. We're now entering into the last 20 minutes of this workshop. So we're going to move into a discussion session. So it's been great to see the conversations happening in the chat, but now's the chance if you want to raise your hand to ask a question specifically to any of the speakers that we've had today. So our 4 keynote speakers, I think you're all still here, and also the 6 early career and mid-career speakers we had in the second part. If you want to use the raise your hand function, or if you want to put the question in the chat, then we can I can either say it out loud or you can say it yourself. Whilst I'm waiting for people to phrase their questions appropriately, I use Chair's prerogative to ask our keynote speakers to reflect on that second session we've just had, with very different perspectives from what we heard in the first section. And I wonder if any of you had any comments about whether they fit with what you were thinking about gaps in your individual areas. I don't know if Chantal or Fernanda or Mattia or Edward has anything particular that they would like to say. Chantal? Thank you.
Thanks. So yeah, I mean, of course, I'm very intrigued by Chesko's presentation at the end. Fantastic. The idea of a condensed framework. I mean, given the huge amount of data that we don't have, if we can limit the data points that we need, that would be incredibly helpful. So I'd love to read your preprint. And are you going to be also writing this up though, from your slides today as well?
Yes, this is a shameless plug moment. So essentially, the CEFACI consortium is going to, wait, I can talk about this, right, Gwen?
Hey.
Yeah, we are going to be launching our website soon. And this, along with other non-journal article pieces, will be available on there. So look out for it soon. I'll send a link to the preprint to the group chat. Super, thank you.
Yeah, just to fill in a gap there, CEFACI is a JPIAMR funded consortium that I'm PI on and Chesko is employed on, and the idea is to try to rank farm level interventions on antibiotic usage against AMR. So we've really been trying to utilize existing data to try and understand what interventions have had an impact and linking data from human and animal health. And once you start thinking about how to do that, you have to start thinking about, well, what's the framework within which we are working and what are the important parameters? That's where a lot of Chesko's great thinking has come from this sort of economic perspective into how we actually try to do that.
And I assume in the paper, do you sort of give the logic sort of justify the condensing. Yeah.
Yeah.
Okay, super. Great, thanks very much, Fresco.
Thanks, Hata. Kuhn, do you have a question or your comment?
Yeah, basically for Chantal, because I like the graph you had of all the things that you want to cost at one point, but I would like to push you a bit, because realistically, that's not going to happen in the next 5 years. So according to you, where would be the most value in addressing it? Because there's so many gaps and so many lack of data literally everywhere.
Yeah, that's very true. So that's going to be basically our first step is, well, seeing what data we have that we think is sufficiently good, and then prioritizing. So, you know, where do we need some primary data collection? So we have a reasonable amount of funding, not huge, but enough to do a bit of primary data collection. But we need to prioritize, definitely. But
yeah,
if, you know, yeah, go ahead.
How do you coordinate that? Because many people are doing something similar, right? They say, oh, there are lots of data gaps. Let's do some primary data collection as well. And is there something to coordinate?
Well, I guess in this case, it's perhaps where we think the costs are going to be greatest. That's where we would focus the primary data collection. So where the data is missing, and we think that those elements are going to have sort of the heaviest bearing on the overall costing, we would prioritize those. But we haven't not yet chosen which areas those are. We kind of have a few hunches, but we still have to really explore.
Yeah, I think that will be interesting to see, because I have my ideas, but I was wondering whether you have very different ideas.
Great, we want to hear them.
There's an element of with what you're doing, Chantal, that is it the current cost of AMR, or is it future costs? Are you trying to understand the burden to the country now to support current plans, or is there any way that you can, you know, look at trends, or is it?
Yeah, I mean, we're going to have to. If we just look at what's going on today, I don't think we're going to get. Well, in some ways, it's not a sufficiently useful picture. I think we have to try to extrapolate a bit into future, and look at different levels of transmission, so that we can basically try to know what we need to prevent. By investing in greater prevention, you know, and control, better stewardship, etc. Without some projections, I don't think the picture is really clear enough as to why we need to invest.
Yeah. Okay. So it's not, this is the current burden you're facing. It's if you don't do something in the future, which is obviously much harder to predict.
Yeah,
right?
Absolutely. Yeah, but I think we'll have to, at least, a bit into future, perhaps on a very, very long-term perspective, but a sort of a short to medium-term perspective.
And so I presume that you're doing some costing of agriculture, but I wonder, having heard Edward's presentation today, whether you are also doing costing or crop data collection, because that was something that I really had no idea. And I wonder if, you know, if you're still on, Edward, about the relative volumes of antibiotic usage in crops. Like, is this something that should be the fourth prong, right? Instead of animals, humans, and environment, we also should have crops, animals, humans, and environment. But
possibly. I mean, the economic analysis is so far out of my area of expertise and comfort zone. But I was, wondering if that would be there would be some way of justifying the massive focus on livestock at the expense of crop agriculture, if we had evidence that actually the volume of antibiotics used in crop agriculture is tiny compared to that in livestock. So actually maybe it's not a massive promoter of AMR in the One Health system. And there's not a great deal of reliable data out there. I think the US probably has the best data sources for the relative amounts of antibiotics used in crop versus livestock. And in that country, I can't remember the figures, but far more antibiotics are used in livestock than on crops, which might back up the kind of less focus on it. But then other countries, you know, there's, as I talked about briefly, you know, reports in places like Thailand about, and India about illicit uses of antibiotics that aren't being, you know, that aren't being recorded and aren't being, you know, they're certainly not being prescribed for that use. So it's a big data gap there about what volume of antibiotics being used in crops in some other countries.
I was very encouraged to see, Edward, that out of like the 5 or 6 countries that answered your questionnaire, I think Zambia and Malawi were both amongst them. So that's fantastic for our work.
Yeah, well, the report is publicly available on the FSA's website, so anyone can go and have a look. And I could post a link in the chat. Actually, would be the easiest. And yeah, there should be more detail on the returns in there.
Super.
So is that something? Is that something you know anything about, Mattia? Like comparing? I mean, I was thinking with your stuff with the point of care diagnostics compared to human health. You know, there's some controversial work from the London School, right, with the UK evaluation about whether point of care diagnostics will actually make a difference, and empiric prescribing is given anyway, independently of point of care diagnostics. I wonder if that's something that you've thought about in terms of animal health. If you're still there. Oh yeah, you have.
No, yeah, I'm still here, of course. Yeah, no, this is a really, really great point. In general, I would say that yes, empiric treatment is still working, but this is I mean in line with the EMA regulation. I mean they advocate for a prudent use of antimicrobial. We should definitely prioritize the antibiotic that are used for humans, and that's why also I mentioned point of care test in my presentation. Of course, it's there to rationalize the use of antimicrobials also in animals. I think it's we need this we need to develop this test to get this to have this test also for animals. They are only limited to human medicine.
Yeah, but that's I mean that empiric prescribing aspect to AMR and the importance of the selection, in some ways it's something that we need to understand as a whole system, right, in terms of we're limiting access in livestock, but then apparently we're giving it loads in crops, and then in other settings we have a problem of access, right, and this kind of push and pull balance is really something we don't have a good handle on, I think.
Yeah, but also the empirical antimicrobial use in animals sometimes is also biased by the fact that we sometimes use clinical breakpoints that are not so available for animals, right? And actually, this is something that I cut from my presentation. But I do also would like to talk about clinical breakpoints in animal medicine, which is something that we are currently lacking, and something that we have to do. We still have to do research on, because we have different animal species, and of course breakpoints are different for each animal species. Thinking about, I mean, I'm working with dogs and with the Pyoderma and stuff. So we also have found some interesting results in the sense that clinical breakpoints that were commonly used might not be actually really exact. So we might also have to go and investigate that from an animal's perspective. Yes.
I'm just seeing some questions coming from the chat. So Leonid is pointing out that if the diagnostics are both accurate and fast, the only remaining challenge would be convincing clinicians. I think from what we know, from what we heard from one of our short talks, right, that changing behavior is still something that's different, even when we know we should be washing our hands, right? Doesn't always happen. So I think it is, as you say, it's a different problem. I'm not sure it's more easily solvable, I think, and the behavioral health, the behavior side of it is really important, right? What did I just read it? Does anyone else have any questions? I'm just trying to. Yes, and then we have Esther who's pointing out that obviously the other aspect of bacteria of behavioral antibiotic usage is that patients often require antibiotics, and I think that's something you touched on too, Corinne, right? Is the sort of need to satisfy the customer to some extent, right? Or the patient or the owner of the pet with the illness.
Yeah, for this was the client. I mean, in veterinary medicine, it's probably even more pronounced. Although you could argue the same for parents with children, that they want something for their child. But.
Yeah. Okay, and if anyone has any pressing questions, I want to wrap up so that we finish on time, unlike the other two sections of today. So if anybody has any questions they'd like to put their hand up.
So if I may just a quick one. So I know that it's been discussed a lot in different venues, but how confident are we at this point that there actually is a transmission, well, a substantial transmission between the animal sector, the human sector, and the food sector? I mean, we had this recent study that showed that there's fungal resistance transmission, or at least as close as we can get to that. But as far as I know, in the bacterial world, we're still not quite there yet. I mean, there's sort of partial evidence, but there's no smoking gun. And if so, is anyone trying to work on that at all, or is it worth working on that? It's just a kind of thinking out loud, really.
Edward, do you want to answer that? Is that hand related?
Yes, yeah, I'll just come in briefly on that. I mean, no, I don't have an answer to that question up my sleeve, but with one of my other hats, I am also involved in the cross-governmental PathSafe program that some of you might have heard of, which is led by the FSA, but involves lots of other government departments and agencies, including UKHSA and the Environment Agency. And that is trying to plan out and then pilot a national surveillance system for foodborne pathogens, but also for AMR. And one of those strands is about identifying sources and routes of transmission of AMR in the green environment, and also it will be looking at AMR in the agri environment as well. So potentially might be able to start to look for evidence of transmission between different components of the agri and the green environment there.
And thanks, Edward. Fernanda, do you want to chip in?
Yeah, if I can just add to that, one way that you are totally right, and this question pops up all the time when you talk about One Health, right? One way that we are also trying to tackle it is not the directly transmission, but actually looking at mobile genetic elements, actually if they can jump from one sector to the other. So not that you have the direct transmission of a whole bacteria or even a whole plasmid, but how these things can move around. So the mobilome part, and see if that can be more relevant, or actually if we can see this kind of transmission in a more fundamental level. But you're totally right, we need much more evidence. And so far, there is no, things are showing the opposite. That seems that this is not such important route as we thought before.
Yeah, I think I would agree with you, Fernanda, that we don't have enough evidence, but you can, obviously you can find many smoking guns. You know, MRSA, we have lots of evidence of transmission from farm animals to farm workers, and these kind of examples in bacteria. But I think the evidence is pushing to more sporadic, right? Than like a big, it's happening a lot, and that's driving AMR in humans. I think we have to really recognize that antibiotic usage in humans is driving AMR in humans. And yes, it's worrying, and we should limit unnecessary, especially human important antibiotics in livestock, but that may not be the main driver in humans. But it's probably species specific. And as you say, we need to think about the potential, especially for the genomic components to jump species, even when they're not human pathogens as well, right?
Yeah, and one thing that we are overlooking, in my opinion, now we also look in the broad at is actually the general population and the pets. So this general contact might be overseen, and we've all focused in the farm because the use of antibiotics there seems to be much higher. But if in the pets is not regulated, you know that for some countries, we kind of overseeing it.
Yeah, great. Okay, well, having had a slow set of questions, then we had a rush, so now it's 2:30, so I'll just wrap up there. I was trying to think of some themes as we were going through the talk there, and I think, as I mentioned, the microbiome came up quite a few times. I think we have to be careful in AMR to emphasize that obviously not all bacteria are bad, and also I think we should be careful to think of not all resistant pathogens as not necessarily being natural components of microbiomes. So it's not necessarily the microbiome is disrupted that we will get AMR, or that if a resistant pathogen is, we would detect a resistant pathogens present in a healthy microbiome, right? There's potentially fitness costs to resistance, but they could be compensated. But I think a theme that's come out today is that understanding what a healthy microbiome looks like in both the built environment, as Fernanda highlighted, and in the gut, and what that means for both disease prognosis and prediction, and even to protect. I think that's definitely something that's come out across that. And then I think the second big theme is the need for intervention evidence. So the importance of context, I think that was really nicely highlighted by some of our short perspectives, whether that's a hospital or built environment, a crisis center, or whether that's Western Africa or Botswana. Your context really matters. We need better data on the importance of context so that we can inform interventions to better use antibiotics, specifically empiric prescribing, because we know that's the main way that antibiotics are used globally, and also to prevent that infection in the first place with better IPC. And so we need data on the size of the problem to focus interventions. And as we showed by our first and our last talks, we also need data to support the case of funding to prevent AMR and to inform national policy design, which is exactly what we started and ended with. I think it's quite interesting that we didn't hear anything about new drug development or anything about genomic tools, which quite often you hear in an AMR day. I wonder whether that reflects the fact that today there's quite a lot of epidemiologists or even a lot of people in the One Health context that are thinking about alternatives to antibiotics and thinking about ways that we can avoid the use of antibiotics altogether, perhaps do better diagnostics or phage, or many things have been raised today. But also it was really nice that we didn't just talk about hospitals and we didn't just talk about high-income settings. So I think that was also really good to hear those perspectives today. So I hope that's given you a new perspective, or at least a different perspective on bacterial resistance than you had before. This has been recorded, so watch out for future communication to have access to the recording. I'd like to say thanks again to all our speakers for really interesting perspectives, and to our participants for the questions in the chat. I'll just pass briefly to Carolyn to wrap it up for today. Thank you.
Yeah, I just love to reiterate what Gwen said and just say thank you so much to everyone who spoke. Thank you so much to everyone for attending. Absolutely massive thank you to Gwen for doing an excellent job of chairing and actually keeping us to time, which is quite outstanding when we're doing such short talks. And then final thank you to Morgan from our events team, who has run all this very smoothly and carefully in the background without anyone even noticing his presence. So thanks again. And yes, look out, there'll be a recording, should go up on the JPIAMR website fairly soon, and we will be writing a report, so that will come out too. But thank you, really thank you. And my goodness, I've got a lot to write in my report.
Thanks, Carolyn. Bye-bye.
Right.
Thanks, bye.